Hi, everybody. It's that time in the program we all really look forward to, which is the industry updates. So we're going to hear from 4 different companies. We try to mix it up, so every year we try to bring in new companies. And this year we will be hearing from Dr. Matt Dyne, who, as you may remember, is at Scripps Research Institute, Florida, and also Expansion Therapeutics. We're also going to hear from Dr. Art Levin of Avidity Biosciences out in California. Then Dr. Laury Mignon, a perennial favorite from Ionis Therapeutics. And then Dr. Qatanani, also from Dyne Therapeutics. So we've got a really nice array of presenters this year. Dr. Dyne had a last-minute complication and had to return to Florida, but we are going to hear from him via live stream, and he's going to be joining us right now. So please welcome Dr. Matt Dyne, Therapeutics. And
thanks, Molly. So today I'm going to talk about work that's going on at which initially started at my lab at Scripps Research Institute, but has been put into Expansion Therapeutics, where we're trying to develop medicines for the myotonic dystrophies. And so you guys are going to hear many different approaches to target the myotonic dystrophies today. And so I want to sort of lay a framework for you guys for how Expansion Therapeutics views these RNA targets. And so what we're trying to do is make a small molecule medicine. So a tried and true small molecule medicine that's not unlike aspirin. It's a compound that you can take as a pill. And then, much like aspirin, the contents of that pill can penetrate many tissues that you want to target or treat. And in our case, that would be tissues such as the heart, the skeletal muscle, or the brain. And now one of the advantages that small molecules have is that many medicines are small molecules, and chemists, like myself, can make many different types of small molecules to fix a problem as a compound emerges through a development pipeline. For example, if that molecule doesn't get into the muscle in high enough concentrations, well, we should be able to make a derivative that can get into that muscle. Or if the molecule, we want it to get into a brain, well, then we believe that you should be able to make derivatives that can cross the blood-brain barrier to do that, all while taking something that you can swallow. Here is the pipeline that Expansion is pursuing today. And so, as I mentioned earlier, but I want to reiterate here, is that the focus of our pipeline is to deliver medicines to patients that have myotonic dystrophy. So our most advanced program is targeting the RNA repeat that causes type 1 myotonic dystrophy, but we also are advancing approaches to target the RNA repeat that causes type 2 myotonic dystrophy. So many of you in the audience obviously are well aware of the DM1 disease mechanism, but I want to just very quickly highlight it here because there's a few points that I want to touch on for the community. And so in type 1 myotonic dystrophy, you have an RNA repeat that's present in a non-coding region in an RNA, or a part of an RNA that doesn't get made into protein. And that RNA folds up into a structure that's shown here. It looks like a hairpin. My mother used to use hairpins all the time. And so when I think about this structure, I think about mom. And so what happens is when these RNAs fold, they become a high affinity binding partner. They bind to, much like a hairpin binds to hair. These structures bind to proteins called muscle-blind like 1 protein. And when this binding happens, that protein is not free to do its normal job, which is to regulate RNA processing in general. And so I want to just make a point here by saying that a lot of what we're doing here in drug discovery is a relay race. And although we are very hopeful that many of us can get to the end of that race to deliver a drug to the patients, we have to be aware that that may just be the end. And sometimes the end is just the end of the beginning, not the total end. And so we want to really make it clear that a lot of this work and this mechanism is built on research from the past several decades by a wide variety of basic and applied scientists that have been working with it for the foundation for a long time, like David Hausman, Maury Swanson, Laura Ranum, Richard Moxley, John Day, Charles Thornton, Eric Wang, and others. And what we also need to be aware of is that the patients have, I think, really accelerated us here, not only through the foundation, but also giving their patient samples to people that I just mentioned earlier so they could do analysis to analyze what's causing the disease. And so the fact that muscleblind regulates hundreds of pre-messenger RNAs is very important. And so last year, you guys heard an update from the Ionis and Biogen team who are developing drugs for DM1. And I want to say that although that initial compound didn't reach it through the pipeline, that the results that Biogen Ionis shared last year were very exciting because they showed that you could test in a patient if biomarkers are being moved. So because of all of that foundation here, I think it's really set a solid foundation for all of the people that are doing drug discovery, and we all need to be very aware of all of their contributions. And so what we're doing in expansion is what we're trying to do is make small molecules that target these RNA repeats very selectively and free bound muscleblind. And the way that we're doing this is we're getting a small molecule to target a structured region in the RNA, that is that hairpin region. And the result is that these small molecule compounds, or these pills that we want to deliver, should be effective. And by effective, it goes back to what I said earlier, is that these molecules have to improve the defects on RNA, they have to improve the defects on protein, and they also have to improve the defects on the phenotype, or how those RNA and protein issues manifest themselves and how a patient might react to the disease. And so as many of you are aware, one of the RNAs whose processing is altered in disease is an ion channel that's on your muscle fibers. And so when you go to have a contraction or relaxation on your muscle fibers, what you do is it's like a battery, is you're controlling flow of energy or electric currents. And when you have a myotonic discharge and myotonic dystrophy, you have too little of a channel that regulates that electric current. And one of the proteins that controls that is this CLCN1 protein that I'm showing here. And so what you can see to the left inset panel is I'm telling you that in a wild type mouse, or a mouse that doesn't have this genetic defect, those mice have 100% of the normal splicing of the messenger RNA that encodes for that ion channel that controls contraction and relaxation of your muscles. And so what I'm showing here is data from a tool compound that we've developed in expansion, is that in a DM1 mouse model that has a knock-in of that toxic RNA that was developed by Charles Thornton and his lab, is that you can see DM1 mice have 0% of this wild type splicing. However, when we add this drug to these mice, we get about 80% of that RNA processing defect improved back to what was observed in a wild type mouse. What we also have known by using this tool compound is that there's something called a favorable PK-PD relationship for drug development. So what we found is that you don't have to, at least in the mice, and we're hopeful that this will translate to a human, if we're blessed enough to get there, is that you don't have to dose this drug all the time, is that you can take a dose on one day, and then you can wait a few days and take another dose. And in between those doses, the phenotype, or the way the disease manifests itself in these mice, is still improved. Now, what I showed you is a improvement in the level of RNA splicing, but that RNA encodes for a protein. And what's shown here in the top left panel is in green, is these are muscles that we've taken from a mouse that has DM1. And what you can do is you can fluorescently stain those ion channels that control the flow of energy through muscle fibers to control contraction and relaxation. And what you can see in a DM1 mouse, it's basically not there. However, below that, in a wild type mouse, you can see that those ion channels are well expressed in there. And then when we treat the DM1 mouse with our drug, you can see that these ion channels are being upregulated, which is what we would expect if the compound is improving defects at the level of RNA and protein. The other thing that we're able to do within these mice is what you can see, and what's shown here are the results for an untreated DM1 mouse. So this is a mouse that has the disease, And what you can do is pinch its hind legs, as I'll show here in this video. And in pinching the hind legs of the mice, you can see that the mouse's legs have a cramp. And that cramp is a proxy for a myotonic discharge. And so you can see that the mouse is not able to relax its muscles in its hind legs when it has DM1. If, however, we take our drug, and this is one of the compounds that we're developing, and deliver it to these mice, you can see that the mouse is much more active in the cage. And then when you pinch its hind legs, it doesn't have that cramping situation. That is, it's able to very quickly, once those hind legs are challenged, able to relax it. And so that's, again, suggestive that this molecule is not only improving defects at the RNA and protein level, but it's also improving it at the level of the phenotype in the mouse. And so one thing I want to, one last slide I want to leave you with before we're going on here is that, you know, medicines, when you go to develop them, have to be safe. And so you really need to make sure that when people have the opportunity to leverage basic science and medical science to delivering a medicine, is that you really need to make sure that these compounds are extremely safe. And so what happens is, what we do is we're developing drugs or design drugs. You make a drug, you test that drug in a test tube, and then once they have a certain activity, you test them in patient-derived cells in the DM1 animal, and then you do this cyclical study to make sure they're safe. And oftentimes, as you go to develop things, you learn new things about the molecule, and you want to go through again and optimize compounds to make sure that they're active and safe. And all of this that I'm showing here within the cycle is things that we've learned from analyzing data that have been published from those people I mentioned earlier that are doing basic science all the way to medical science. So what we really need to do to make this work is we need to have patient-derived cells, know how they work, know how the defect manifests themselves, be able to use an animal model to be able to see how these compounds work in an animal to improve defects, and then put these molecules into a series of very robust testing. And so I think that's one of the issues with drugs. Don't happen overnight, because it's very critical that as you go into a developmental pipeline that you ensure that those compounds are safe and effective. And so I just want to summarize here is that expansion is developing small molecules that target RNA repeat expansions broadly. And our focus is on delivering on the promise of that basic research on DM1 and DM2 to try to get a medicine. And so we're focusing on both of those diseases. How we're able to do this is we have a platform where we can make small molecules that target structured RNAs. We have a wide variety of other tools that we can use to affect these targets. For example, we can cleave RNAs with small molecules. One of the foundational approaches is that we're using classic small molecule approach like aspirin or ibuprofen that you can take in a pill. And we believe we have a lot of expertise in this area that we can leverage from not only chemistry, so I'm a chemist, but I do some biology, but also in biological sciences and also in the medicine of DM. And much of what we've learned on the back end in drug development, and I want to reiterate this because I think it's important, is Biogen Ionis's work really, I think, set a foundation here to understand how biomarkers can move in these patients. And we're hopeful to use not only the foundation of that, but also the basic science to advance therapeutics for both DM1 and DM2. And so I want to thank you for taking the time to listen to me. Thank you.
Thank you, Matt, wherever you are. That was really nice. That was a really nice presentation. So Matt's working on small molecules, which is a very exciting potential therapeutic approach, and it's one of the rarer approaches that we see for myotonic dystrophy right now. We also have people in the room that are working on gene editing therapies. Jean Yo, who unfortunately had to return to California, is working on gene therapy approaches. So there are lots of different therapeutic approaches that are being used right now to target myotonic dystrophy. One of the ones that's probably been in play the longest is also the one that has probably the largest number of companies currently using it, and that's the antisense oligonucleotide therapies. And you've heard that phrase before. Ionis and Biogen have been working on that therapy since quite a while and making great progress. So the next three speakers we're going to hear from are all ASO companies, and it's exciting to see all this expansion in the space, as it were. I want to invite Dr. Art Levin from Avidity Biosciences up to talk about the approach that they're taking.
So I would really like to thank the organizers. It is a pleasure to be here to talk to you. I've learned a great deal over the past couple of days. What I'd like to talk to you today about is a mouthful. Antibody oligonucleotide conjugates, and there's my disclosure. I'm the Chief Scientific Officer at Avidity Biosciences. So that's a lot of words. Let's talk about what those words mean. Basically, what we're doing here is we are combining two very powerful technologies together, and I'll work through it. Let's start with the oligonucleotide part, because that's where Molly left it off. So an oligonucleotide is a small piece of RNA or DNA. It has been designed to bind specifically to a region of a gene, in this case, the region of the toxic mRNA that's associated with myotonic dystrophy. So we make this perfect match for the gene. The oligonucleotide will then find that gene, and then like north and south poles on a magnet, they'll bind together. And the act of those two, the oligonucleotide drug binding to the messenger RNA, that toxic messenger RNA that Matt was just talking about, causes the toxic messenger RNA to be cleaved. And that's really the mechanism of action that we're talking about here. So I've been working in the field, as the people at Ionis know, for almost 25 years now. It's really gratifying to be able to come to you and talk to you about something which is so close to the clinic. The issue is that we can develop an oligonucleotide for almost any disease-related RNA or any disease-related protein. But the hard part has been getting that oligonucleotide into the muscle cells or into cells in general. So we're taking advantage of another very powerful technology called antibodies in order to take kind of a Trojan horse approach to moving oligonucleotides into cells. Our cells don't want to take up foreign genetic material like a little piece of RNA. If it does, you're going to get mutations and all kinds of crazy things. So we've evolved over the past 5 billion years not so that our cells don't take up oligonucleotides that might be associated with viruses or bacteria. So we're trying to fight against 5 billion years worth of evolution. So we've discovered this technology to overcome that barrier. And on the next slide, what you can see is this is our Trojan horse approach. So I know it doesn't look like a horse, but it works like one. What you can see here is that we have that Y-shaped molecule is called an antibody. An antibody is a specialized protein that we've designed to bind to a specific protein that sits on the surface of muscle cells. Antibodies are pretty magical. So if there were 1 billion molecules in a haystack, and the transferrin receptor, which is what we've aimed this molecule, was one of those molecules out of 1 billion, it would find it every time. So this is a very powerful way to recognize a specific protein that sits on the surface of a cell. So in this particular diagram, what you're looking at is a cartoon. It's a little bit complicated, but let me walk you through it. What you're looking at here is a cell, and at the upper right, you can see our Trojan horse. Our Trojan horse has that little red tail on it. That little red tail actually represents the oligonucleotide drug. Here's our Trojan horse outside. It will find the receptor that's shown in brown here. The cell then thinks it's taking up a specialized form of iron, because muscle cells need a lot of iron. But instead, we've actually fooled it into taking up our drug. And our drug is then engulfed by the endosome in a normal pathway where cells are constantly engulfing nutrients that they need. And then it's released, and it can get loaded into an enzyme complex called RISC. It's the RNA-inducible silencing complex. The Nobel Prize was given for this technology about 12 years ago. Once the oligonucleotide is loaded into RISC, and you can see it here actually, here's in red, you can see that's our oligonucleotide that's loaded into RISC. Here's the north-south kind of polarity binding to its target site. Once it does that, that enzyme complex is going to destroy the toxic messenger RNA. And that's really what's shown on the next slide, which is a diagram that you've already seen. Matt Disney just showed you a very similar diagram. The Ionis people, as Matt said, demonstrated last year quite nicely that if you knock out this gene, if you make this gene go away, make this messenger RNA go away, you're going to get improvements of splicing, and you're going to see the changes that Matt showed in those mice. More importantly, Laurent and others showed that, in fact, you could do it in patients. And unfortunately, they were at such high dose levels that they really couldn't go on with that study, and they didn't get as much drug in as they needed. What I want to show you today is by taking advantage of this Trojan horse mechanism and by taking advantage of this RNA-inducible silencing complex, we can actually deliver a more potent oligonucleotide and do it better because it's targeted delivery. So the remaining slides I'm going to show you are actual data from a monkey study. So we're very far along on Matt's circle of evolution of drugs. We're at the final stage where we're doing the final stage of testing in monkeys. And what you're looking at in this slide is an example of a dose response curve. So in this study, monkeys were given single doses of intravenous doses of either 1, 2.5, or 5 milligrams per kilogram of a drug which is targeting the DMPK gene, the toxic messenger RNA. And what you can see is that a dose of 1 milligram per kilogram produced greater than a 50% knockdown. If you measure the concentration of drug in that muscle, so if you take that biopsy and then measure the concentration of drug in that biopsy, it's 1 nanomolar. That's 1 part per billion with a B. With respect to the Ionis compound, they were trying to achieve a concentration of approximately 1 micromolar, which is 1 part in a million with an M. So what you can see is that not only have we created targeted delivery for our system, but we've done it with a molecule, with a payload. Our Trojan Warrior is even more powerful than the Trojan Warrior that Ionis was using. By the way, I was an Ionis employee for 12 years. I love those guys. This is science, though. The next slide is a really interesting one because this one will hopefully affect you directly. So what you're looking at now is the knockdown of gene expression in 2 different muscles, the quadriceps and the calves, the gastrocnemius. And this is a single dose that we gave on day 0. And what you can see is that out at day 42, 6 weeks after the administration, we've still reduced the toxic gene. So we have a really nice long-lived response after a single dose. We would predict from these data that we'll be able to dose this drug once quarterly. The next slide is also very important to you because we spent the whole afternoon talking about cardiac defects and we talked about breathing problems. So we can also change gene expression with the toxic RNA in heart and in diaphragm, and that's what I'm showing you here. So again, this is our dose response curve. A dose of 1 milligram per kilogram produces greater than 50% knockdown. By the way, these data were taken on day 42 after administration. So this is 6 weeks after a single dose. And you can see that our dose of 5 milligrams per kilogram is producing greater than a 75% knockdown. Now, you know from the patient literature that someone with myotonic dystrophy that has 50% more CUGs than another patient has a much less severe phenotype. So again, the CUG dose is important. Here we can lower the CUG dose 75% from starting in a normal monkey. The next slide is for the scientists in the audience, and I apologize to those who aren't. So I'm going to do science talk here. We're talking about half-lives. I'm going to go really fast. We're talking about half-lives of the drug in muscle. That's what's shown on the left. You can see the half-life of the drug in total muscle, homogenate, is about 2 weeks. If you measure the drug that's actually incorporated into RISC, that RNA-inducible silencing complex, the half-life of the drug in RISC is 2 to 3 months. So there's no magic that our drug works for 6 weeks or more. It's actually sitting in that enzyme complex stabilized so that you get long-term activity. So that's why I can say with some degree of confidence that we'll be dosing this drug probably quarterly. So just to wrap up, I know I've spent probably more than my amount of time here. We've developed the technology. for using antibodies to provide a means of moving oligonucleotides into cells and treating muscle diseases like myotonic dystrophy. We also have a muscular dystrophy program. Our payloads are very potent. We can produce long-lasting effects with our payloads, and we're working with the utmost speed and diligence in order to move this technology forward. And of course, the only thing you want to hear is, when is it going to be in the clinic? And that's the last line here. We expect to be in the clinic. We have a year's worth of toxicology data to do ahead of us, and getting through FDA with the initial application. We expect to be in the clinic in early 2021. I'd like to thank my coworkers. There's nothing that makes a small company pull the oars in the same direction as getting everybody in a racing shell, and that's what we're doing. That's what we've done here. So here's the gang anxiously looking to help you out. And then the last slide is, here's some reading for you to do. If you're interested in the technology, there are a couple of papers that are out in the literature with my name on them nicely. And they're actually edited by the brilliant editors in the New England Journal for readability, so they are actually readable at a kind of a college biology level. And then this is the dream paper that I'm looking for. Right, I have to press that button. The goal, of course, is to have this paper, the successful treatment of myotonic dystrophy with an ASC.
I think we're a big fan of Art's publications. That was really nicely done. This is not easy science, and I think he did a very nice job of explaining it. I am really pleased to invite Dr. Laury Mignon from Ionis Pharmaceuticals up to the podium again to hear what they're up to, both in terms of the different programs that they're working on.
Well, hi, it's a pleasure to be back, and I'm always happy to be here. And I have to say, it's quite a new era for being here and having had two speakers before me bring up the Ionis Biogen collaboration. So this is really quite exciting. But I'm going to be talking today about tackling the CNS, and I'm actually not really going to show any animal data, human data. It's going to be a little bit of a different talk. So first, for the people who are new coming this year to the foundation, I just wanted to let everybody know where I was from. So I work at Ionis Pharmaceuticals. As you've heard before, the company has been built 30 years ago, and it was really built to develop a new platform of drugs, and these were RNA therapeutics. And so Matt and Art have nicely explained to you kind of the foundation, or how our drugs work. And so I was kind of hoping they would do that, so I could go through this slide a little easier. But really, what we're targeting is we're trying to use these little pieces of ASO that we call, so they're little pieces of DNA that we put together, and we can easily target an area on the messenger RNA that we want to, because that's an easy code to find. And as you all know, the issue with myotonic dystrophy is those repeats, because those repeats will then bind these muscle-blind proteins that Matt and Art were talking about. And if you bind these muscle-blind proteins, they can be released to do the things that they normally would do, like regular splicing. And so without muscle-blind, you get aberrant splicing. And you get that in the brain, you get that in the muscle, you get that in the heart, you get it everywhere in every tissue. So what these little pieces of ASO do, they bind outside of those repeats. They bring in an enzyme, and then that enzyme will break down the messenger RNA. You release those little red dots, and now muscle-blind can go do what it's supposed to do. So that is how our drugs work, and that's how our drugs work no matter where you would give them, right? If you give them systemically with an injection like we did in our trial, you can affect the muscles or any tissue there. And then if you wanted to go to the brain, you would actually have to give them intrathecally, because our drugs do not cross the blood-brain barrier at this point. So just as, again, both Matt and Art mentioned before, we did a trial. The trial was negative in the sense that we couldn't go on with the molecule because we didn't have enough potency of the drug. So we would have had to go to a much higher dose than we can give So we went to doses up to 600 milligrams, and just to kind of give you an idea, because 600 milligrams, you don't know what that is, that was basically 3 1 ml injections. And so people had to get injections, you know, on the day of injections, 3 injections, and that's pretty much the most you would ever want to do. And so we had to go back to try and find a drug that was more potent. But what we did find is that we did change splicing pattern. And so we looked at a bunch of different splice events. And just to orient you on the slide, sorry. On the bottom here is before treatment, as you can see, and after treatment. And basically what this data shows was splicing events taken from the muscle. Patients were giving out muscle biopsies before treatment, and then there were muscle biopsies taken 2 weeks after treatment. And so 0 is person who is not affected, 1 is severely affected, so the higher the dot is up on that line, the most affected the patient was, or the most affected the splicing was in that patient. And so again, like I said, this was before treatment, after treatment. And so you can see that there was really not much effect. The dots stayed all nicely on that line, except once you got to the higher dose, you had those 2 little guys, so that actually did better after treatment because they didn't stick on the line. So we had an improvement. We could see that the mechanism of action works, right? So that was good, but like I said, we had to go back to the drawing boards. But what that also allowed us to do, though, it allowed us to think about the CNS. So we have had a lot more experience in the CNS since the time we started our muscle compound in myotonic dystrophy and now. And because the modality is the same, we figured now we also want to try and tackle the CNS. And like I said, we've had programs in spinal muscular atrophy, Huntington's disease, Alzheimer's disease, ALS, and so we feel much more comfortable being able to go into a disease giving intrathecal injections. But that is also one of the problems. We have to give intrathecal injections. And so that will be what the next trial would be in myotonic dystrophy if we were to target the brain. So one of the things as we're trying to move forward to trying find a drug now for the brain, we're also trying to see what endpoints we could use. And the FDA wants to make sure that drug developers really understand how a patient feels, functions, and survives. That is a term or a sentence that they use a lot, and so we have to focus on that from beginning of development through to approval of the drug. And so we did that by really looking at what was out there already in terms of what do we know about CNS symptoms. The Myotonic Dystrophy had a really nice patient-focused drug development meeting that they led in 2016. They also did, and they published a voice of the patient, so we could pull on that and see what kind of information we got there on CNS symptoms. They also did the bringing the patient voice of the CNS targeting drug at one of the MDF meetings, which was also extremely valuable. I remember we were there and listened to the patients, and it was really quite fascinating to hear what they had to say. And then there's also reports like the Christopher Project, who covers all the symptoms. But again, we were focusing now on trying to better understand the CNS symptoms. Are there endpoints out there that we can use? Are there maybe symptoms where another disease has an endpoint for it? Because we need to be able to go in the clinic, measure the symptom, and then see if we have improvement. So what we've been doing as we are screening for our drugs and as we're waiting for our tox studies to happen, we are focusing now on really trying to decipher the CNS symptoms that are important to all of you. And so we put out a survey this summer, and I want to thank Myotonic Dystrophy for sending out the survey. So we put out a survey where we're trying to understand really the symptoms, and I'm going to show you a little bit of data that came out of that. We haven't had time to really fully analyze it. We just got the results about, a week and a half ago. So we are trying to pull stuff together to be able to share that with you today. So in only 3 weeks, we got 258 surveys answered. So kudos to all of you guys for really answering that. The survey was written so that it was really for DM1, DM2, and CDM patients. We had 191 patients respond, 143 of those were DM1 patients. And then we had 67 caregivers respond. And you see the numbers there. I can't see the numbers so far. We also had we had 4 CDM patients respond and caregivers, but there was not enough data right now for me to be able to show that. But we will publish this survey, and so hopefully you'll get to see all the results. So really at a high-level assessment, I think the first question we asked is kind of like your overall symptom, what is most burdensome, and just to orient you on the slide, again, really should learn how to do this. So we asked, the first question was really general. We asked, physical, CNS, gastrointestinal, cardiovascular, and then the dark reds is most bothersome, and as you go up is least bothersome. So obviously the physical symptoms were still mentioned as being the most bothersome symptoms for the patients, but you can see that CNS and gastrointestinal were really, close second, and then cardiovascular less so. Now, when you ask the caregivers, for the caregivers, the highest ranked symptoms, though, was CNS. And this is something that I think we've heard before and we've seen before in other reports, so this was not really surprising. We then went on to try to better understand the different CNS domains, and so we asked specific questions on cognition, fatigue and daytime sleepiness, apathy, anxiety, aggression, obsessive-compulsive disorder, and communication. And so I'll show you a couple of those here. So all the next couple of slides will be oriented the same, so I'll go through this in detail so that you can read the next ones easier. I'm going to try to point to this side for now. So on the left you have the answers from the patients, on the right you have the answers from the caregivers. And basically we asked questions about, do you feel like you have impaired decision making? And the answers were never, sometimes, or almost always, right? So the never are the gray bars, sometimes the blue bars, and almost always the pinkish bars. So I think you can see that it was pretty similar results between the patients and the caregivers, except that I would say the caregivers in general for these cognitive symptoms thought that it was closer to almost always, or more almost always than sometimes. And again, the questions here were impaired decision-making, challenges with mental exercises, trouble finding words, was it hard to concentrate, hard to remember things from the past, like childhood memories, hard to remember things to do in the future, going to appointments, things like that, or hard to remember recent events, a party last week, or so forth. And we would always put these examples, we put those in the questions so people would understand what we're asking about. In terms of fatigue and daytime sleepiness, similar setup here of the answers. And again, I think the concordance between patients and caregivers was pretty good. The few items that I thought was interesting, where the caregivers rated mainly never, was actually symptoms that really the patients would know better. And it was, I wake up before it's time, or, I have trouble staying asleep, or, I have trouble falling asleep. Obviously, those are symptoms that are closer for the patients. In terms of apathy, I think, again, it was pretty similar between both, except for maybe caregivers thinking that the patient showed, lack of energy or motivation on a daily basis was maybe stronger seen from the patient's caregivers. And then the last one I wanted to show here, just for a matter of time, is anxiety and depression. I think, again, maybe there was just, caregivers felt that maybe the patients felt more anxious, but in general, I think, patients, and we've heard it in the sessions before, patients are anxious about, what's to come, or maybe feel depressed about, where they are at, and the things that they will not be able to do anymore in the future. So in conclusion, what we are really trying to do, as I said, as we're screening for drugs for the CNS, is really trying to understand, to better understand the CNS symptoms, and even when we say cognition and executive function, is to really try to nail down what symptom is important to you, and then also to understand, can we measure that symptom, and can we have an endpoint that we'll be able to use in a clinical trial? Because at the end of the day, we're going to have to show some improvement in a clinical trial. So like I said, the survey does confirm previous work that has been done. I think it expands a little bit on that as well. And we will be publishing the results of this and putting this together in either publication for the MDF or poster presentation, but this was really the first time we even had a chance to ourselves look at the results. And just wanted to finish with what Dr. Janet Woodcock at the FDA always tries to tell us, is that we know the patients are the ones that know the disease the best, so we have to listen to the patients and then take that information on in terms of trying to develop the better drugs we can for you guys. And before I finish, And Molly runs over here. I just also wanted to thank Molly for all that she's done. She's been a great collaborator to work with for industry people. She's kept us honest, so you guys definitely need to give her a round of applause, and we just love her.
No more of that. Okay. I'm delighted to introduce Dr. Mo Qatanani from Dyne Therapeutics. Dyne is based in Boston, Massachusetts. They're also working on ASO therapies. They're doing some exciting work, and I'm happy to have Dr. Qatanani join us on stage.
Thanks, Molly. Good afternoon, and I recognize I'm the only one between you and cocktail hour, which is not a popular place to be. First, I want to thank the previous speakers for actually laying the ground for what I'm going to talk about to you today. I want to thank Molly and the organizers for inviting us to come here and present to you. And what I'm going to do is tell you a little bit about Dyne, about our technology, share with you some of the exciting data that we are excited about, and then tell you where we're going with developing therapies for DM1. So Dyne Therapeutics was founded with the main focus of discovering and developing life-transforming therapies for serious muscle diseases. Our primary indication and disease is DM1, but also looking on other diseases. The premise of Dyne and the challenge comes from the fact that you've heard already. There's numerous muscle diseases, genetic muscle diseases, including DM1, where the cause and etiology of the disease is known. We know that that pesky RNA that's in the cell that's wreaking havoc in the cells. So that part of the challenge is solved. We also do know there exist technologies like the antisense oligos that have been pioneered by Ionis and others that can attack these RNAs and get rid of them. And in that case, reduce the burden of the disease or even reverse the disease. And yet, we don't have any therapies on the market for DM1 or any of these other diseases. And that's very challenging. It's very frustrating for drug developers. It's very frustrating for patients and their caregivers. And very frustrating for all of us. And the reason that we don't have a therapy is that these oligos, when you put them in the system, they go everywhere. Most of them go to the liver, to the spleen, very little to the muscle. And what we need to have is targeted delivery of antisense oligos, like we've previously heard. And this is where Dyne comes into place, where we're going to take these oligos and deliver them to the muscle so that they can get rid of the RNA and then reverse the disease. So the technology itself, it's three components. The squiggly things on the sides of this Y-shaped things, these are the antisense oligos that can be siRNA or antisense single-stranded. Linked to this Y-shaped things, this is an antibody that targets a receptor that's highly expressed on the muscle. And the way you can think about it is that this Y-shaped thing is a shuttle that's going to take the antisense oligo to the muscle so that it'll get in and degrade the RNA and reverse the disease. And then once you have all of that together, you have to go through the studies and then manufacture it for clinical trials as well as hopefully approved therapies. So before I show you the data, there are several stages in drug discovery, and it requires a lot of different inputs from different stages and different departments. When you have different development candidates and you want to pick the best one out of them, you start testing with cells. So you put them on cells and you see how they react. The next step is to test them in small animals, and these animals can be either mouse or rat. And the next step is to test them in bigger animals, and these are the monkeys or the non-human primates. After that, you go into humans clinical trials, and just to level base, sometimes you hear phase 1, and these in general are in healthy volunteers, not always. And when you hear proof of concept, this is in patients. And the closer that you get to humans, so the closer that you get to the monkeys, the higher the likelihood of the drug actually working. So in this slide, I'm going to show you one of the data that we generated in mouse in this case, where we injected these animals a single dose and then took down the mouse after 7 days. And if you look at the data, and keep in mind now that we're looking at DMPK, that pesky RNA that we need to reduce in muscle cells. So looking at the data, and we looked at a variety of different muscles, almost 14 to 15 different muscles, skeletal, smooth, and cardiac. The black bar is the placebo, though, injected with PBS. And you see that's at 1. The red bar is the naked oligo. That's an antisense oligo that we injected as a control. And you don't see changes. The gray bars are the antisense oligo linked to a generic protein that doesn't bind to our receptor. And you don't see any change. But the focus is on the blue bar. So the blue bar is that antisense oligo linked to the Dyne therapeutic antibody. And now you can see dose-dependent decrease in DMPK at low dose at 55%, higher dose 77%. And you see that across all the muscles that we looked at. Skeletal, gastric, soleus, you see it in the heart as well as the diaphragm, which is very important for the patients. So moving from mice, we moved to non-human primate into monkey. That's the next stage. And this is a study where we did an IV injection and then take down after 14 days. And we took more than 14 different types of muscles, focusing mostly on muscles that are affected in DM1, like the TA and the gastrics and others, and the heart. And when we looked at the data, you can see also consistent and robust knockdown with our Dyne conjugate versus the red bar, which is the free oligo. So 62% in the gastric, you see significant knockdown in the soleus, in the heart, as well as the diaphragm, which is important. So this table summarizes some of the data that we have. Some of it I've shown, some of it I haven't shown. So we looked at potency of the antisense in muscle, knocking down the RNA in both mouse and non-human primates. We looked at muscle tissue specificity, so we looked at knockdown in muscle versus other tissues. Tolerability, which I haven't shown, at least in single doses, now in multiple doses, that seems to be safe. We looked at everything, including liver function, CBCs, et cetera. Durability, something that I haven't shown today, but we do see durability beyond 28 days, which gives us the option of dosing once monthly or even once a quarter. As well, in mouse models, we also tested this in the MSXL mouse model that actually showed engagement of the human RNA knockdown. So as far as our plans and where we are, right now we're in preclinical studies, selecting our development candidate, getting into IND-enabling studies. These are the studies that will allow you to get into humans. We're supporting the natural history study to actually get us more information about biomarkers and establishing our clinical protocol and plan. And we'll be looking at mechanistic and functional biomarkers for our BLA and hopefully approval. Last, I want to reiterate our mission, which is focused on developing therapies for serious muscle diseases, with DM1 being our primary indication. I would encourage all of you to actually go to our website and look at it, where you'll see the story of Joaquim that's featured here. And it's really his story and stories like him that are at the core of the urgency that we feel to develop these therapies and move forward with them. And I want to thank all of you for the previous sessions where you actually shared your stories. I salute you because it takes a lot of courage to share these stories, and it's really motivating. And you guys give us a reason and meaning to our work every day. So thank you. And I want to thank Molly too.
Thank you. All these thank yous. So now everybody is completely conversant in ASOs, right? You've had 3 presentations on ASOs and how they work, and you feel very comfortable talking about them. Me too. I would like to get into our Q&A. You guys had lots of questions this year. It was very exciting. We tried to triage them down to buckets of questions for which we had the most. And we have an outstanding panel, as usual, of people to answer your questions. So if Darren Monckton, Dr. Darren Monckton, Dr. Laury Gutman from Iowa, Dr. Darren Monckton from Glasgow, Scotland, Dr. Missy Dixon from University of Utah, Dr. Jacinda Sampson from Stanford, Dr. Laury Mignon from Ionis Pharmaceuticals, back at ya, And Dr. Katie Eichinger from University of Rochester. We like to be geographically diverse. If you guys could come up to the stage here, and if we could get a wireless mic, that would be great. To hand back and forth. Like that one right there would be good. So we have a few questions here. I've decided to just kind of hand them off to people. And we're going to start with Missy. Because she's not up here. That's what you do, too, when people are at dinner and you ask somebody a question when they're chewing. Yes. So we had a number of people ask this question, Missy. How do I handle apathy? You can chew first.
Is this on?
It is.
Is it a caregiver asking? Is it a
It's a patient asking.
patient asking?
I didn't tell him the questions ahead of time.
Well, I'm just trying to reconcile my thoughts about apathy, and frankly, I think apathy, as we're calling it, is really executive function problems. So I would recommend you can do executive skills training, something like that, or try and pinpoint, what exactly you're feeling apathetic about as you see it, if that's the case, and maybe further flesh out, what exactly the executive function or dysfunction is, and then go from there.
Yeah. Missy is doing a lot of psychiatric and behavioral work at University of Utah and has done some presentations for us here at the
Can I
foundation.
just interrupt again, because Darren reminded
Oh,
me something
yes.
I said earlier. So if the feeling of apathy is coming on because tasks are difficult for you, or it just seems overwhelming, the idea is that, maybe breaking it down into more easier chunks.
Mm-hmm.
And so that could be a caregiver task, or, an example would be, I have laundry to do today. I have mountains of laundry for my 2 children and my husband, and I can't imagine doing all of it today. And to say, okay, I'm just going to work on, one load at a time, today, and maybe one this afternoon. So breaking it down into more manageable tasks.
Perfect. Excellent. Thank you, ma'am. This one is for Jacinda. Does foot drop happen to those with myotonic dystrophy type 2?
It can. So I think, when we originally learned about myotonic dystrophy type 2, we imagined that it was proximal. That was one of its original names, that it was proximal myopathy with myotonia. But as we are finding more of it and looking harder for it, we are seeing that there is a greater spectrum of symptoms. And so I think foot drop can happen with it. And there's, as one of the things we learned about this morning with the neuroimaging, is there can be asymmetry, and there can be a great variety in the age of onset and severity, and is it weakness, is it stiffness, is it pain? And so I think there's a lot of diversity in the presentation in myotonic type 2.
Excellent. This one's for Katie. And this is large. What are the best types of exercises for someone with myotonic dystrophy?
So that can be a range of different things. I think the most important things is it's something that people enjoy doing, because if it's not something you enjoy doing, you won't continue to do it long term, and it's important to continue to do exercise throughout your lifetime. It is also something you need to take into what your abilities are, and so obviously there's a wide range of abilities, and so I can't just say what the best thing is. Certainly in people who have more distal weakness, so weakness at the ankles, sometimes the best exercises are to build and work on the muscles, or not necessarily build, but to work on muscles that are more proximal. So the core muscles, your abdominal muscles, trunk muscles, and your hip girdle, as well as your quads, can be muscles that can be less affected by the disease and therefore can be more amenable to doing some strength training and exercise. As far as different types of exercises, working in the water, doing cycling are often nice exercises to do with muscle disorders, because they can respond differently. Water kind of supports weakness, but also gives resistance to the stronger muscles. Cycling, the foot is often locked into the pedal, and so you can use more of those proximal muscles. So I think there's lots of answers. The answer could be large, too.
Okay. Perfect. This one is for Dr. Monckton. Totally for you. Do the advanced CRISPR-type approaches target, or would they target the disease at every stage?
So obviously, the CRISPR approach is what we call a gene editing approach. So it's a way that we could potentially cut the repeat out of the DNA sequence. So in myotonic dystrophy type 1, we could cut out the CTG repeat, and in myotonic dystrophy type 2, we could theoretically cut out the CCTG repeat. And if we were able to completely remove the repeat, we think that would completely eliminate the disease process in the cell in which we did that. And in cells in a test tube in the lab, that's been done, and it appears to be very effective. The big challenge is how we do that method that works very well in the lab and actually apply it to an intact human individual. And there's a couple of ways we can think about that. So one is to take out cells, fix them in the lab, and then put them back in. But actually, with a condition like myotonic dystrophy, that's going to be really challenging. We can't replace every muscle cell. We can't replace the brain cells. So there's going to be limited ability for that approach. So the other approach is to try and put this machinery that edits the DNA back into cells in the person. So there are different ways that can be done that. So one way that's being commonly done now in research models, in animal models, and in some other diseases it's starting to happen now, is to put the machinery into viruses that can deliver the machinery back into the cells. And again, there's a lot of work needs to be done before that's going to happen in human trials, and particularly in this condition where, again, we would have to get it into lots of different cells. But theoretically, it could be applied at pretty much any stage of the disease. Obviously, a person would have to have a diagnosis, first of all, for that to be possible, but there's no reason, once we work out how to do that delivery, that it couldn't be applied to any stage. But it's a massive challenge to overcome that delivery issue. And again, there's also issues. Again, this is one way I think what we're doing here is cutting out the DNA. And that's great if we cut out the bad DNA, but there's always a risk that we might cut out a good piece of DNA. And there's obviously a side effects issue with that again needs to be resolved before we could think about doing that in clinical trials in humans. So there's a lot of interest and a lot of excitement about that technology overall, but there's quite a long way to go before that's going to be a realistic treatment in this condition. But MDF is funding some research in that area, and there's a lot of research going on in that area, specifically in the myotonic dystrophy. So again, it's something that's happening, but it's going to be a while yet before it's a realistic treatment option.
This is for Laury. Is the drug approval process going in a similar direction here as it is in Europe? Or are some drugs being approved there but not here, and vice versa? And maybe you want to just address that.
Yeah, so most of the times, so it's tricky how you want to answer this, because I can answer it from the drug developer perspective. So most of the time, a drug developer will go both in the US and try in Europe at the same time to get a drug approved. And most of the time, as we're working on that clinical program, and especially before we go into phase 3, we actually try to go talk to the regulators at the FDA, but then also the ones in the different countries to get approved or to get alignment with them on, are you okay with this trial design? Would you be okay with this primary endpoint and things like that? So we do try to get, global alignment. Sometimes that doesn't happen. Sometimes the two, regions don't necessarily agree on the primary endpoint and things like that. I think we've seen both drugs that got approved in the US faster than drugs that got approved in Europe and vice versa. So I think it is kind of tricky to really answer this on a whole. I think for rare diseases, regulators globally will be more open to different ways or novel approaches. So it's a little tricky to...
It depends.
It depends.
Okay, this one's for Darren. What is known about why the congenital form is passed maternally and rarely paternally?
Yes, so that's a tricky question. So again, what we know is that most congenitally affected children have an affected mother, but there certainly are cases of paternal transmission of the disease where an affected father has a child with a congenital form. Some of that is down to the dynamics of what is happening in the germline of the parent. So one of the main drivers of whether a child is likely to inherit congenital form of disease is the number of repeats inherited. So typically it's high hundreds or even thousands of repeats. And it seems to be that females who have a couple of hundred repeats are able to produce eggs that contain high hundreds or even thousands of repeats, that then gives rise to children with congenital. Whereas males, most males, for reasons again we don't really understand, can produce sperm that have hundreds of repeats, but not frequently the high hundreds or thousands of repeats. So that's one, the sperm that are being produced are just not capable of generating congenital. And again, we don't really understand why that is. But there's also a couple of other factors. So again, there may be that there is something specific about not just the number of repeats, but something about what we call an epigenetic change. So the DNA can change in both the sequence, but can also have a special modification called methylation. And it's possible that that is different when the DNA gets passed on from a male and a female. But there are also a couple of other effects too. So one is that males with the adult onset form of the disease are more likely to be infertile than females. So males in that middle generation have fewer children, so there's a lower opportunity to have congenitally affected children as well, and that's at least part of the explanation as well. So it's a combination of a number of different things, and not all of which we know why they're like that. So we know they're different, but we don't know why they're different.
Excellent. All right. This is a question for Laury Gutman. How do I approach a family member about encouraging them to get tested for myotonic dystrophy?
Well, so there are different reasons why people want to get tested or don't want to get tested, and so that's the first thing to think about is, even though you may have been diagnosed and you might be concerned about a family member, or maybe you're just concerned about another family member, that person may not want to get diagnosed, and so it's an individual's decision about whether or not they want to get tested, and I'll just put that out there for starts. But one of the things that we do in our clinic that is you could ask for from your physician is something that we call the family letter. So we have a letter that's written, it's a pretty standard letter that talks about what myotonic dystrophy is and what are the symptoms that people with myotonic dystrophy might have. And it starts out by saying, someone in your family has been diagnosed with myotonic dystrophy, and you may want to consider whether you are affected by this, and then it lists out some of the symptoms, and then it talks about who you might want to go talk to about potentially getting tested. That's a nice way, and then you can send it out to all of the family members, whether you know that they're affected or not. That's one of the things that some of our patients do. They sit down and talk with our genetic counselors and then talk about their concerns, and then very often they go home with what's called the family letter.
That's perfect. And this is a very similar kind of topic, and this is for Missy. What are some considerations that parents can keep in mind for determining the right time to tell children or test children who may or may not be displaying symptoms?
Well, that's an individual decision. And so I think having an understanding of why do you want your child to be tested? Why do you want to tell them? Are they mature enough to handle it? What would you gain? Are some questions that I would ask from telling them now versus at another time. And those are things that I think are important. Also, how mature are they to understand the complexity of it? Are you experiencing symptoms and that's why you want them to know, or is it just another family member that has some and you're curious? So, and then also, I always think knowledge is power, but that's how I feel, and that's my family system. And so I do think it's individual to your family system and your family and how you would want to manage that.
Yeah.
And I don't recommend saying it at night, right before bed, or when everybody's tired or hungry. I think you would really want to plan for that conversation and make sure that you have time to answer questions and to deal with possible emotions that may arise.
Yep.
Can I add something to that? I'll start by adding something to that, and then maybe Jacinda too. I mean, one of the things is if your child doesn't have any symptoms, in our clinical setting, we discourage that unless there's something that you can treat. So until there's if there's a cure, then that's another story. We aren't there yet. But if you're just concerned about whether or not your child has it, but they aren't displaying any symptoms, testing may not be the best idea. And the key thing to do, I would recommend, is talk with a genetic counselor about what the pros and cons of that are. But the other thing is, if your kid is having symptoms, getting a diagnosis sometimes helps in the school systems and getting them the services that they need by having the diagnosis. So that's another thing to consider. Jacinda?
I think you covered a lot of the points I wanted to make. And one is that if there is something that makes you suspicious that they are having symptoms, particularly if it's affecting their education and development, then that's an indication to test. Another thing you want to talk about with your child or your young adult or your family member is not only when do you want to know, if they want to know, but what do they know now about myotonic dystrophy? What do they think about it? What do they know about it? How do they feel about it? Because that helps you prepare for that discussion, because they might have very real fears about why not to test at a particular time, whether it's work or relationships or getting insurance, that there might be real obstacles there, that they may want to be tested, but this might not be the right time for them. And it's because that it can affect healthcare and prevention, we encourage that if there's a family member at risk but is not interested in being tested, that they ask their primary care physician to do an EKG annually and say, I have a risk of a heart disease, a heart problem in my family. I would like an EKG to be part of my annual physical. And then you can feel safe about at least monitoring for that if the decision to test is delayed or put off for other reasons.
That's really good.
Oh, and family planning can be a factor in a question. And so
Yep. We could do this all night, but we
could.
won't.
One of my favorite topics.
These guys know what they're talking about. So I'm going to ask one last question. This one came up a lot. I'm going to ask it of Jacinda. Does obstructive sleep and central sleep require different therapies?
Yes and no. So how you diagnose them is all through a sleep study, where you can look at the sleep patterns and your breathing patterns in sleep. And obstructive sleep apnea looks different than central sleep apnea, but the way that we treat that is the same, with non-invasive ventilation to help support your breathing. And one of the things that came up in the talk on sleep earlier today is that not only can, depending on your machine and your settings, can it support your sleep with different pressures when you breathe in, giving you more air and a lower pressure so you can breathe out more easily. You can also set it so it has a backup rate. So if there's a pause in your breathing, it's like, it's been a couple of seconds, let's skip a breath. And so that can be designed into your sleep support. So knowing that is, I think, a useful thing. And I know getting used to sleep apnea treatments, whether it's central or peripheral, can be difficult, and trying it during the day to get used to it. Behavioral therapies, if you have claustrophobia, are really important. Are there other things that lead to anxiety relating to it that can help it be better? Is it noise? Is it the mask is too tight? Is it leaking and blowing in your eyes and making your eyes dry? Is it making your mouth dry? Do you have too much saliva and it makes you feel like you're going to cough or gag? All those things are important to talk about with your pulmonologist so that you can make it work for you. Because treating sleep apnea, if you can tolerate the machine, it really does benefit your health in so many ways. With your breathing capacity, it's less strain on your heart, it's more rest. You're more rested in the morning. And no, it's not a cure for all the daytime sleepiness or fatigue, but every little bit helps.
Perfect. All right. So you guys have been troopers about hanging in here for 2 solid days. Hopefully you got a whole lot of really interesting information. So we wanted to give you a little treat. I'm going to invite John Fitzpatrick up. Is John in the room? Oh, there he is. I'm going to invite John up. He's got some fun things for you.
It's time for the 2019 Myotonic Dystrophy Family Conference Challenge. Let me hear it. I said, let me hear it. It has been an amazing 2 days. But now, it's our opportunity as our community, that we're all connected by genetics, as we've learned, to pass the hat. And so our goal by the end of the evening is to raise $100,000 through this challenge. I have some great news that thanks to an anonymous donor from one of our community families, we've got a head start of $35,000. And then John Brecka, can you raise your hand? So John is one of the founding board members, one of the early pioneers. Wave it again, John. They didn't see you the first time. Pink shirt or lime shirt. John retired as a lawyer, and now he is crafting handmade reindeer. The first 10 people that give $250 or more will get one of these. There's now 9, because I'm bringing one home to my son, Jack, for the Smurf box, Kristen. That's what we'll be doing. But I know Molly didn't like the acknowledgement before, but we've got to say a few words about Molly. So it's been almost 8 years, and I was fortunate to lead the board search committee when we hired Molly. And in 2011, at the Clearwater Family Conference, which was also jointly done with IDMC, that was Molly's first time coming here. And for many of us, the first time you walk in a room of a couple hundred people with myotonic dystrophy, it is a very powerful moment in your life. Molly had been through the interview process, but this was the first time that she saw it. And I just want to thank and challenge all of us to write a check today for Molly and to honor her almost 8 years of service. So Molly, this is for you. So Molly did not want to come up here and brag on herself, so I asked her, what are the 3 things as she's leaving that she's most proud of in terms of her legacy. First are the 45 companies that were here at this conference today. Thanks to Molly's leadership, we are the hub and the network for pharma and biotech across the United States. Incredible. Second, I was really struck by the brave stories of our over 50 AARP, DM1 and DM2 folks that spoke today. And this past year, we had 7 clinical guidelines or resource that are dropped that are now available on the website. One for DM1, one for DM2, one for congenital, one for cardiac, one for respiratory, going to school in K-12, and Social Security benefits. Thank you, Molly. And then last, but certainly not least, where's Ralph? Where's Ralph Northen? Ralph, raise your hand. So Ralph, this year, your old employer, the United States Department of Defense, has contributed $3 million to research for myotonic dystrophy, thanks to advocacy by Molly and me. So can I ask all the board members and the staff to stand up? They all have those little square things. So I want everyone to reach in to your wallet or your purse, take out your credit card or your checkbook, and please join Kristen and my family in making a contribution to honor Molly's service. We'll be back in 5 minutes.
Okay, so we're going to take a change of pace, and we're going to select the winners of the raffle for the free trip to San Diego next year. I believe we've got 3 different prizes, so get your tickets ready. We're going to stir it up really good. Okay, Jack, you want to do the first one?
186100. 186100.
All right.
One 186-154. 186-154.
All right.
The last one is 186026.
Thank you, Jack and Ben Kelly. Winners, if you can see the registration guests. Okay, we've got about 4 more minutes on the fundraising. We are almost at our $70,000. We're at $67,350. So we just got a little bit more, and then we'll continue after dinner.